Okay, great. So, welcome to the second session. I just took the opportunity to put up my collaborators here. As you can imagine, it takes many people not only to gather data on so many children across several countries, but also to analyze many different image types. And I like to rely on the expertise of very skillful collaborators for each type of analysis. Okay, so let's go back to that last slide. I think this is a pretty good topic for discussion. So we have this problem that the literature is scattered with many small studies that report significant results that are probably not reliable. Only a few of them will replicate, and of course we have the wrong incentives. We have the incentives in the research fields are to publish a lot of papers, to publish them quickly, and of course in the journals with as high a possible impact factor. And so all this does not encourage people to gather large sample sizes and to do very stringent corrections for multiple tests and so on and so on. So I'd like to open the discussion. What do you think we could do to try to possibly change the research practices, change the incentives such that everybody would from now on publish only studies that have a fair chance to be replicated? Ideas? I mean, obviously those who have read the Ioannidis papers have a head start on this, but
I didn't read, so yeah.
One idea would be like pre-publicate. I mean, you just submit your research idea and the criteria that you're going to use to the journal, and they kind of pre-accept it, and then you just follow those things instead of choosing things post hoc, I guess.
Yes. So here you're trying to address the problem of flexibility in data analysis and post hoc decisions, such the problem of choosing the regions of interest a posteriori, depending on what you have observed in the whole brain analysis. So yes, one solution is so-called pre-registration, such that you submit in advance the design of the study and also including the analysis plan. So like I plan a new study on the plenum temporale in dyslexia, and I write down the whole procedure, the whole imaging parameters, and the whole pre-processing parameters, and then the analysis plan. And I say my region of interest is the plenum temporale, therefore I will allow myself to test the group difference using a t-test or whatever without correction on the plenum temporale. And then I carry out the study, and then you are referring to submitting this to a journal. So like, for example, Cortex is a journal that encourages submissions of pre-registration such that you submit only the introduction and the methods of the study before conducting the study. They peer review it. If it is accepted, then you conduct the study and you resubmit the paper, adding the results and discussion sections. And provided that you have followed the method that you have declared in advance, they will accept it. Okay, there will be no further judgment of importance or relevance or whatever in the second round. Yeah?
Yeah, I was just asking equally if it's a null result.
Equally if it's a null result. Okay, because that provides you currently one problem is that there's no incentive to publish null results because you know that it's going to be published in less good journals than if you have a positive results. So you leave the null results in the drawer and you move on to, or either you p-hack in order to find a positive result, or you forget about the null results and you move on to the next study that is going to be published in a better journal. So if you want to change the incentives, you have to make it equally worthy of publishing a null result. And so having the guarantee of being able to publish it in a given journal, no matter what the results turn out, is a good way. I would like to add that this is the journal-based pre-registration, which is a pretty good solution. Not many journals offer that at the moment, but Cortex is a good one. There's also the possibility of pre-registering the study in an online public database that is not necessarily connected with a journal. It's just out there, and once you've submitted it, it can be checked later. So when you submit your paper in any journal, you can say this study was pre-registered, and you can check the pre-registration document at that link. Okay, and then reviewers can check that and can check that you follow the analysis plan that you had declared. And of course, this does not prevent you from doing exploratory analysis. I mean, I have plenty of brains on my hands, and I don't know exactly what sort of, I mean, there are probably differences between dyslexics and controls that exist for real, and that I wouldn't have the idea of hypothesizing in the first place, and I want to have a chance of discovering them if they exist. So I think exploratory analyses are worthy too, but they are submitted to different rules. So in the pre-registration system, you pre-register your analysis plan for your a priori hypothesis, and then you are free. I mean, you have less stringent corrections for multiple tests. This does not prevent you from doing more exploratory analysis after that, but you declare that they are exploratory, they were not motivated a priori, and therefore you have to apply all the stringent corrections. Simon, you had a question?
Yeah, I was wondering if anyone's had experience with the cortex route, because I was under the impression that there is a second review. And
There is.
It's a light review, so it is possible that if you had results that weren't very
No, but at Cortex, they explicitly state that they will publish the paper regardless of the nature of the result, as long as you followed strictly what you had declared.
So then they're reviewing it on the how interesting the research question is, and so long as you have a solid study design as well.
Yes, in the first round of review, these are the criteria, the soundness of the research question and the appropriateness of the methodology. And then they check that you actually did what you said and that you conducted the analysis properly, of course, and that the discussion is okay. You may be asked to revise the discussion if you conclude too far beyond the data, of course, but they will not reject only on the grounds that you have a null or uninteresting result.
So I think it would be interesting to know how well the whole process is going in
practice,
how
yes.
people are submitting on this route, and how
Yeah.
it would be nice to
I haven't tried it yet. Dan?
So you mentioned published null results. It came as a surprise to me to discover that both PLOS 1 and what's it called, science or scientific reports, measures have very high impact factors. So in point of fact, if you have a result which is technically solid, you can publish, in principle, should be able to publish those papers in those journals, and they will, on average,
Yeah, I think the impact factor of +1 was inflated in the first 2 years, and then it's going to decrease steadily until it reaches some floor, because there's no reason it should remain high, given their publication policy.
Scientific reports, which is even more recent, but the last, when someone brought it to my attention, I looked, +1 was 3 point something or other, and scientific reports was 5 or 6 or something. So if you care about that.
Yeah, but I think, scientific reports or Science Express, they will go exactly the same route. They will have a few splash papers in the beginning that drive out the impact factor, and then it will be downhill from there. So I think it's kind of self-correcting, if you will. But I think there's another problem. I mean, in principle, I really like the idea of the pre-registration. But what we see is that the reviewers are totally overburdened. And that creates another, sort of basically doubles, the amount of reviewing that needs to be done. And I think you need a complementary measure that is really bringing down the number of papers.
So the one that I think is possible is you do an exploratory study and then you do a replication study, and only when you've done these two together, you're publishing.
Yeah, so, I mean, okay, so that's another parameter indeed. I mean, we say that we need to increase sample size. So obviously, if we increase sample size, we're going to run fewer studies with the same limited resources. Or if we are compelled to run replication studies as a condition to publish, then also, of course, this is going to decrease the number of papers. Now, of course, in the cortex system...
Is that a problem? I mean, I think we're faced with increased number of exponentially increasing number of papers. Now, every year, there are 100,000 papers in the field of neuroscience. And that's way too many that anybody can handle.
Absolutely.
The whole problem is the fact that we have to care about the career of the young people. They need publication, but the whole system is unsustainable in the long run.
I agree entirely.
But perhaps a simpler step, I mean, if we don't want to go to the pre-registration, is just, I think we discussed this, to do a power analysis, to make it mandatory. And if a study doesn't fulfill certain criteria, it doesn't matter if the result is significant or not. You say, well, your sample size is too small on average, and you say...
Yes, so power analysis is another step that is part of the picture, and that indeed should be mandatory. So you should know in advance how many subjects you need to test in order to have a good chance of discovering an effect of the size that you think is the real size of the effect. Then of course it may be difficult to predict in advance what the size of the effect is, but at least you should have a good guess at it.
But you could use that to automatically reject these studies with 10 or 12 people, because even if they are, like you said, a lucky, if you can show that the power analysis...
But it's not easy if the power analysis was not pre-registered in the first place, because you can always make up a story based on your data. Look, I've got this huge effect size, so this means that I had enough power to discover it. I mean, when the power is calculated based on the data that you have collected, because there was no prior data that would allow you to have a good estimate.
Sorry, at least in Nijmegen, every study has to be, for all the PIs, has to be presented, and we don't accept a study they don't get measurement time if the power analysis is not then done, and if the power is not sufficient. So that is total control, and that I think we can all do.
I mean, that's very difficult. I mean, I'm all for it, but in an electrophysiological study, say, how do you know, even estimate the power, if you don't know the effect size? You do like 10 different analyses. And you just don't know because it's so sensitive to, say, signal to noise ratio of a given experiment. So actually putting your money where your mouth is with respect to power analysis is easier said than done.
Yeah.
Because when you know you're going to do, inter-trial phase, God knows what analysis you're going to do, that's very fickle, and you don't know in advance.
But you can bet that it's not going to be a large effect size.
That you can bet. But I wanted to ask.
And moreover, let's say because often in these cases you've done more than one study, so at some point you have some experience based on pilot data and so on, from which at least you can derive some estimation of the effect size that you can use for your power.
Certainly, that's how you do it for grants based on your past studies, right? So this is the
that's also what you should do for your.
But I want to ask the people who work with animals, especially with monkeys, because so I mean, for us it's very easy to. Well, not very easy to talk about replication and power and stuff, but how is it for working, let's say you train a monkey for 6 months to do a task, then you record your 80 cells, does that ever get replicated? Is the notion in a way, that's very...
Yes, you do 2 animals, that's the standard.
Yeah,
that's the standard.
Yeah, that's the standard.
It always gets replicated in 2 animals.
So, I mean, and are we supposed to be, I don't know, are we supposed to be impressed by that or worried?
Is there
I mean, it's not so clear, right? I mean, yes, that's why you see 2 monkeys or 2 birds, but I'm not sure how it
feels. 2 birds isn't enough. This is utterly peculiar. It's a different standard. You could never publish it. You should never publish it just 2 birds.
And in mouse studies, you have to have, it's like 10 mouse degrees.
That does.
But should you also adjust for the fact that you published a study already with, say you're working on a data set that you've obtained from some monkey studies or some human brain imaging studies, and then you go, I want to now look at this data set in relation to another hypothesis. Of course, that then, should that be adjusted with respect to the fact you've already done, examined one? Do we have to adjust our whole scientific careers?
This is getting philosophical, I think.
I mean, in neuroscience, I think one thing is what are you looking for? How many cells, for instance, do you measure? You know, how reliable is your finding for one monkey? And then you're looking whether you can find that pattern in another monkey. And then I think, given the ethical implications, I'm not so worried. But if I look at behavioral observations, where the behavior between individuals, variation of the behavior between individuals is humongous. I mean, as I talk about, we have some smart monkeys and we have some really daft monkeys, you know? And then we only have 9. And I think, I mean, perhaps within the next decade or so, what we have to do is we have to bring together all monkey researchers worldwide and pool the subjects and come, you know, have standardized ways how we test them, or standardized protocols how we observe them in the wild, so to at least that we can, you know, aggregate data from different sources, because I will never be able to house 400 monkeys, you know.
Yeah, and I mean, there is, it's
Yes,
two monkeys because it takes a long time. That's the reality. So if you wanted to make 10 or 20, then no one would ever affect it.
And are the effect sizes good? He's kind of saying these biology studies?
That's a practical argument, but that's not the...
Well, it's not like that's not
Yeah, because
Not a principle, yeah.
I mean, let's say if you want to get reliable data, then it takes the time it takes. You can say, well, 2 monkeys is for all kind of practical limitations, but if that's not, because then we can also say here we continue running small-scale studies. It takes a lot more time if you want to run 200 MEG subjects than when you want to have 12. And if that's the consequence, if that's what we need to do in order to get.
At the very least,
There's a room full of, you know, graduate students and postdocs who ultimately want to see
so that's
and there's an interaction between those two which cannot be.
yeah, that's exactly why the system is unsustainable in the long run.
I'm not defending it, I'm merely explaining.
Should we ask the participants what they think about it?
Can I maybe add one little thing because of the replication as Julie already said. Replication with we're doing the electrophysiology in monkeys, so it's 2 monkeys, that's right. But we're not measuring just once these cells, so we're doing this over and over again over weeks to record from single cells, so that at the end you have, let's say, 1,000 neurons you record from, so that this is also some kind of a replication, so that the monkeys did several sessions, and in all these sessions, or in most of these sessions, you find these neurons in this monkey, right?
Yes.
Yes, but your statistical inference allows you to say something about this particular, so it's a fixed effect kind of effect, right? If you want to make statements about...
Yeah, you can only talk about the 2 monkeys.
Yeah, right.
Yeah.
So you can say, well, I can make theoretical inferences, but you're not allowed to make statistical inferences to the population.
well, then a minimal improvement on that would be to work in tandem with another lab, and each time you find a result, ask them to run a replication study on their 2 monkeys. And then if you have consistent results across 2 and 2 across independent labs, that's already a bit stronger.
But that's a problem that's not allowed actually. So we cannot do replication studies with monkeys.
Well, that will have to change, I suppose.
So that means that the notion of the thing is that means the notion of replication or trust in or truthiness of those data are then the incidental replication. So suppose we take, I pick your favorite area MT or something. Right? So people have recorded in that for dozens of years, and it turns out that Bill Newsom gets this kind of finding, and someone else gets this kind of finding, and it turns out that there are good convergences, but there's never a real replication because the monkeys were trained differently for different amounts of time. So what are we supposed to take away from that? That we just believe it because it's neurons, because there's a more elementary in some sense? Well, I mean, it doesn't give me confidence, right?
No, I think what may be true is that in some of those studies, the effect sizes are huge. And so you see it in 2 or 3 individuals, and that's enough to convince you that it's something reliable in most monkeys.
No,
but these are huge effects, like
in these monkeys, I would say it's a strong effect in these monkeys.
It's not randomly effects that you can generalize to the population, right?
That's what we do. You can't generalize.
So the point is, if you on the one hand say there's a huge behavioral difference between these monkeys and so on, we can't, and at the same time we're assuming that, well, but at the level of the underlying neuronal responses, they're exactly the same. How do we explain then the fact that there's some different...
Yeah, but they're in completely reduced paradigms. I mean, you know, Stefan Treu, he has moving random dot patterns, you know, or you have like one frequency that you're testing. I mean, the paradigms are very different that you observe or that you do with neurophysiology compared to what we do cognitive testing in the lab, or even if we go out in the wild, and then it's completely, you know.
Isn't there a problem also in like collaborative studies that the people get rewarded? There's lots of people doing the work, but not everyone's going to get rewarded because so first authors are going to get the prize, and then lots of people put in a lot of that time as well, end up not getting the benefits of the work.
Yeah. The problem about replication is you've got all the ethics problems. So you apply to do your study with these two monkeys, and you can't then just say, well, and now I'm going to do it with these two other monkeys because you follow all the other.
I know.
But you know, the ethical standards are things that can change, and in fact, they have already changed in the clinical domain. So there was a time when people would say, oh, you know, you can't include 100 patients in a clinical trial because you waste their time when they could have a good treatment. Or it's unethical to put people in a placebo control group because they should have the best treatment available. And then the outcome of this debate was that, well, it's unethical to involve patients in research that do not allow you to draw reliable conclusions. So first you should design research that allows you to draw reliable conclusions, and then we can discuss the ethics. Otherwise it's no use to do the research. And so now it's accepted to have very large groups of patients and to have placebo control groups, and people have found ways to make sure that people who are on the placebo group get the proper treatment afterwards and so on. I mean, there are several ways to solve that. I suppose that for the monkeys, this will have to go in that direction as well. If you, unless you can really prove that the results that you obtain are reliable. If that's the case, fine.
It's never going to happen, or at least not long. I don't see it happening given the reality of where non-human primate research is.
Well, already pre-registration, even without increasing sample sizes, pre-registering exactly what you're looking for in those monkeys and so on. This will diminish the risk of false positives. This would be already a good move.
Is that there's a great deal of good science to be done by Ecole.
Yeah.
And you have to be able to
Absolutely.
be able to find things that you could never.
No, but then
Are there cases of spectacular failures, like in your field, say? Are there cases where there was a really wonderful claim made based on, let's say, single unit data, which then turns out 5 years later, say, actually, this is completely false?
There are interpretations of data that turned out to be incorrect. There are questions about whether certain manipulations have this effect here or not there, that are still open questions, but I can't think of any. When Mark Kanishi deafened white-crowned sparrows, it affected song development, and every single species that has been looked at, every single Aussie passerine bird that has been looked at since, the same result. It's a very compelling.
Yeah, but this is not exploration.
Exactly.
This is a priori hypothesis.
Exploration is followed by replication. And I think that
when he was doing those experiments originally, it was in a lab that was against doing skin in anything. And he developed, through his own dogged determination and belief that this could work, he developed the technique to doing the removing the cochleas. It's much more complicated than what we know to do nowadays, to deafen the birds. And he persisted, and he got that result, and it was very much like that, against a background that, we really should never cut the skin, break the skin. We should never be...
Yeah, but being against the mainstream is not the same thing as exploration. He had a very strong prior hypothesis and he tested it. He could perfectly have pre-registered it. Exploration is a different thing. It's like you could explore zillions of different things and you do it, and then you find one thing and that's what you publish. I think it's fine too, but then you should be required to state exactly everything that you have explored so that people who read your analysis have an idea of the likelihood that you would have obtained your finding by chance. But that does not occur for the moment, but that's what we should move towards.
You can't pick and choose which results you report. That's acceptable.
In general, I believe this will happen.
That people report everything they have explored?
Honest on what they are exploring?
No, no way. No way.
No. But there's a difference also, I think. Sorry, you want to go?
So part of the incentives will come from the high-profile journals. In clinical studies, if you want to publish the results of a clinical trial in Lancet, New England Journal of Medicine, British Medical Journal, and so on, Nature Medicine, the first thing you have to say is where is your pre-registration. If you have not pre-registered your clinical trial, you're not even allowed to submit your paper, and you're going to go to a very low-grade journal. So everybody who runs a clinical trial has to pre-register it, and that helps removing a lot of the junk, filtering a lot of the junk. And so I don't see why our journals wouldn't do that or have requirements about replication or power analysis. I mean, they could very well do that. If the good journals do it, then everybody will follow.
But there are still limits. Like, for instance, when we started the study on the guinea baboons in Senegal, so we had some ideas how they could be organized, how their social society might look like. Then we went there and we did a proper description, and I think we have a relatively good database now. We are not able to replicate this ever, because when we go somewhere else, the conditions will be so different, that we will not conclude anything. So we can only ever talk about this population and say, okay, over the last 10 years, we find the same pattern over and over again in different individuals. They behave like this. But that's all I can say.
Well, there are some areas where the data are intrinsically scarce, and you have to live with it. But you also have to take the results with a pinch of salt.
Well, these are descriptive data that hold for that particular site and that for that particular population. That's fine, but that's also clear then. So once you want to make general statements about all baboons or wherever they are in the world, of course that's not legitimate. But when it's there are lots of descriptive studies where you can say, well, describe this what we find.
But then the paper got rejected because we didn't test any hypothesis. And I said, well, in this context, it's completely ridiculous to test hypothesis because I could have come up with 30 different hypotheses, and I present you the one that turned out.
Yeah, but that depends on the journal, what their kind of thing is that they want to have. So then you have to go to a journal where they accept these kind of things.
Yeah, I did in the end.
Okay, I think we should move on to something else, but Simon, do you have an important point?
I was just going to ask maybe a show of hands from the participants as to how many people would feel like if they're about to start a new study, let's say their PhD or their postdoc in a new phase, would they be do this pre-registration? Who thinks that they'll do it with the next study and who thinks they won't? So who would?
Wait, who will do it? Who will do it? I've got to write down the names. We're taking names here, doing it tomorrow.
Of course, it's difficult because it's much easier said than done. I mean, I'm giving you this talk, but I've never pre-registered any of my studies, okay? And I hope I will in the near future. But it's difficult because there's no incentive to do it. So we need to reform the systems to make sure that everybody has the right incentives.
I just wanted to add that there's a small conflict with the fact that the agencies that give funding, one of the main criteria is the number of articles that you have, right? And they filter out people without even looking to what people are doing, just based on the fact that, okay, you are working for 4 or 5 years, you have this number of articles, you are out. So what are the risks? Are we the ones that are supposed to start changing the system so that we suffer the consequences? Or are the ones that are on the level of PIs, the ones that are on top of those in the faces of those agencies?
Yeah, we agree that this is a vicious circle, and you cannot escape it, of course, by yourself.
Yeah, but I mean, you can convince us step by step to go in that direction. But I think it's your cohort's position is actually to change that culture fundamentally.
Well, with our help.
I'm sure you said that you know the sort of pressures that we are in, right? So that's the difficulty to be able to change this from being outside and in a completely, I mean, that's something for us to try and figure out with the people from the people like you.
But you also have to take into account that if you pre-register your studies right at the beginning of your PhD, this offers a kind of guarantee that you'll be able to publish them. Much more so than if you don't pre-register anything. Like you could pre-register all your brain imaging studies with Cortex right at the beginning of your PhD, and you know you'll have some papers by the end, no matter what the results turn out to be, which is not a guarantee that many students have at the beginning.
I'm skeptical of whether every PhD student who wants to send their papers to Cortex, the pre-registered, will get accepted at that point.
Well, Cortex alone will not suffice, but...
Yeah, but for a dissertation, it's particularly advantageous because you have to plan a dissertation. Your thesis advisors have to approve it. You have to have an analysis department. You have to have, some idea of power analysis. So all of these things are done already, so it's to your advantage to pre-register.
Yeah, if you do it right from the beginning,
You can't do it at the beginning. It's also, I think that works more in the European model than the North American
well,
model.
no, at the time when you plan the experiments, it's relatively costless to pre-register them. Of course, as soon as you're already engaged in a project, it's already too late and it doesn't work anymore.
Yeah, I'd just like to say that it's really easy to pre-register, for instance, on the Open Science Foundation. So you just deposit a document in which you put the motivation of the study, the method, and the analysis. So the cost is very small. Like you spend 2 hours writing your pre-registration once you decide what you want to do. And it means that for all your generation, if you start doing this, you have this good practice of putting it in writing in advance. And then when you do your analysis, if you don't find what you expected, at least you remember what you were expecting at that time. So it's also a frame of mind. It's really good practice to start doing these things. Even if it won't buy you anything immediately. It's good that the young people start doing that. And in our lab, Franck Ramus, the students are forced to do that actually. It's where we've decided in the master's program, they have to do it. So.
But I don't think it's only the bottom-up thing, right? If we are all starting to, if we all, so now what happens is that the people who are less strict about, how the standards they have, they get more publications and they get further in science, right?
No, I agree. It can not only be bottom-up. It can be bottom-up for whatever is relatively costless. And for the rest, it has to come top-down, like from the top journals and so on. Does this still work? No, I don't think. I think the mic does not work anymore.
One more general comment on the question. I mean, we are talking about how to get into high-impact journals and which requirements we should have for high-impact journals. But I mean, there is this tendency to generally go more into open access and open peer review, and I wonder how long this whole system of this journal-based system will last anyway over the next generations of young researchers and with the uprise of the internet. So I wonder if this is...
That's a whole different discussion.
Yeah. I mean, the problem is that it's a self-sustaining system, but yeah, it's probably going to be replaced by another system that has better incentives. But I have the feeling it's going to be slow.
I mean, the German Science Foundation, for instance, they decided that you can, when you apply for a grant, you can only submit 5 papers. In this way, they wanted to bring down the numbers of papers that people publish, so that you send in your 5 best. And the researchers rioted. They hated it. They said, no, I want to submit 10, 20, not everything. And the reviewers, what they do is they go online and they check the website and look at the rest of the publication record. Yeah? So there is this sort of schizophrenia, or the same in ERC panels, where they also had a limited number of papers that people could submit when they did the grant application, and then everybody went online and calculated Hirsch indices and whatnot. And so I think there has to be, because it's a social dilemma, right? But there has to be an institution that implements certain limits and regulations. And one, at least at our university, I think it would be great if they would do away with that rule. You have to have 3 papers for your PhD.
Can we continue this maybe after? Because I would still like to hear this.
Thank you. Okay, so Yeah, now I have another set of slides, not a very large set of slides, which I put together very recently because I was inspired by the other talks, and so I'd like to share these thoughts with you. So we're still sticking to the brain structure level, but of course the whole point of studying brain structure in dyslexia or in anything else is also to try and connect it with brain function on one side and with genes on the other side. Okay, and so how do we best do it? So first, I'd like to show you some examples of mappings between brain structure and something else, because after all, there aren't so many examples of really successful mappings. I mean, I've shown you what I think works best in dyslexia. There's not much left that I would really bet money on. Now, let me mention this. It may seem completely silly, but there's a correlation between IQ and total brain volume, and it's a very well established effect. It's based on a meta-analysis of plenty of studies, and the correlation is 0.33. Okay, it may seem entirely uninteresting because who cares about IQ and who cares about total brain volume? Yet, I think this is the strongest relationship between brain structure and and cognitive function that we know of. I mean, there's nothing stronger than 0.33, perhaps by an order of magnitude. So we should not dismiss it too easily. And I can even show you one illustrative scatter plot, which, as you can see, is split between men and women, because it turns out that there's a sex difference in total brain volume, which does not translate into a sex difference in IQ. Here I put the graph for verbal IQ because we're talking about language. It's true also for performance IQ. As you can see, the correlation between, and here it's white matter volume, because as brain size increases, of course the thickness of the cortex does not increase, the surface of the cortex increases, but much of the increase in volume is taken up by the white matter. So the white matter volume is what is most relevant here, and there's this nice correlation between white matter volume and verbal IQ that holds both between males and between within females, but of course the two groups are offset on the total brain volume dimension only. Okay, is that pretty clear? You were shaking your head. Do you know of a stronger relationship? I think we can't find anything as strong as that.
The only thing that worries me is, but it's a single case thing, that you take Einstein.
Yeah, sure. I mean, it's a correlation of 0.33. In a correlation of 0.33, you will have all sorts of individual counterexamples, as usual. Yeah.
I don't know if you checked this, but did they also show in that paper if there's a correlation of size and height?
Height. I don't know in that study, but in fact it has been there have been studies of whole brain volume where they adjusted for the total height, and this definitely does not explain this relationship. I mean, there is a very small correlation between height and IQ as well, but it's not of the same order of magnitude. Okay, that's one that I think is interesting. Here is another one that is pretty intriguing. It's a very recent study by colleagues in Paris. They are the guys, you know, who were trained in a neurospin team, and they use this software Brain Visa where they automatically label the sulci, which is also another type of anatomical variability that I think is pretty interesting. And so here they've noticed that the cingulate sulcus can come in two varieties, a single-stranded sulcus and a double-stranded sulcus. You can see the second branch here. Okay, and that's in each hemisphere. And so here they're looking at whether the two hemispheres agree in the variety of cingulate sulcus that you have. And they have found that there's a relationship between the symmetry of the anterior cingulate sulcus and cognitive control as measured in a Stroop test. Okay, so these are the reaction times in the Stroop test, and these are the error rates in children. Okay, these are relatively young children, preschoolers. Okay, so that's again interesting because these are relatively young children. These are properties of the brain, again, like the plenum temporale asymmetry, that are relatively unlikely to move much after birth or after a few years of life. So they're probably not the outcome of differential experience. And maybe they are able to explain some individual differences in cognitive skills. Now, of course, one caveat is that the sample size for that study is extremely low, 11 and 8. So it remains to be seen if this is ever replicated. But I think if it is, then this is a pretty intriguing result. Yeah.
Sorry, and the different ACC pattern, did they because in some cases you'd have the double parallel
yes.
pattern on the left hemisphere and other cases in the right hemisphere?
So symmetric stands for 1-1 or 2-2. Asymmetric stands for 1-2 or 2-1, and I don't know. I mean, the problem is that they don't have the power to split into those who have 2 on the left, 1 on the right, or vice versa.
Because there are studies, Pausch has done studies in which they've examined the prevalence of this double parallel pattern, and it's more often occurs in the left hemisphere than the right hemisphere. They've also done studies on that. So
could be. I mean, generally speaking, I'm showing all this because, I mean, this is the very broad topic of the link between structure and function. I mean, I'm convinced that there's, of course, a link between structure and function. There is a wide variability in cognitive abilities. There is wide variability in brain anatomy. I mean, you just look 100 brains, and they're all different. And we have only a very weak understanding of what those differences in shapes and size mean and explain in terms of cognitive ability. I think this is a major area where we could discover plenty of interesting things, but for the moment we have very few clues. So this is one. This is another one about the cingulate sulcus. This is about the length. So this is, the paracingulate sulcus. This is about its length. Okay, this is an example of a long paracingulate sulcus, and this is an example of a short paracingulate sulcus. And in that study, they have shown it to differ, the length of the paracingulate sulcus to differ between controls and schizophrenic patients, and also within schizophrenic patients between those who have hallucinations and those who don't have hallucinations. So those who have hallucinations are those who have the shortest form of the paracingulate sulcus. The other neurospin group, they also have a different result about the shape of the superior temporal sulcus, explaining different varieties of hallucinations in schizophrenia. Here, as
you ask the question, because you could say, well, I mean, I can derive multiple brain measures and correlate one, and so is this the one, or is this, if they have looked at something else?
Did they pre-register?
No, of course they did not pre-register. So you are right. Here.
The point is, what, I mean, let's say, what's the explanatory? Why would the paracingulate sulcus and its shape or length be the factor that is related to schizophrenia and not something else?
Yeah, you're absolutely right. Here, I mean, I looked up at their introduction, and they seem to be building on a small literature suggestive of the implication between this sulcus and cognitive control and schizophrenia and so on. So they did not start from nothing, and there seemed to be some relatively reasonable a priori hypothesis. But you are right that otherwise we can look at zillions of things in the brain, and 1 times out of 20 we get a significant result, and that's what we publish. So we get exactly the same sort of problems that we were talking about, that these are very interesting results, but we should believe them, especially if they were pre-registered, right? Or if there was a very clear a priori hypothesis that is not debatable. We've done our bit with the CELSI, I don't know if we have a reliable result. I'm showing you what we have. In fact, we have a. Oops, sorry. We have a triple interaction between group, sex, and hemisphere in the depth of the central sulcus. The central sulcus is here in red, and we have this triple interaction. Here, I have not plotted the triple interaction because it's impossible to grasp, but I've reduced it by computing the asymmetry index of the depth of the central sulcus. So this removes the hemisphere factor, and we only have group and sex factors. So you have females in orange and boys in blue, control females here, and dyslexic females there, control males here, and dyslexic males there. And you can see that the difference between the groups is in the opposite direction depending whether you're looking at girls or boys. I don't want to sell you that result, although it's very highly significant for several reasons. For one, we have no idea how to interpret that. I mean, it could be that we see a difference in boys and not in girls, or the opposite. We have seen that before. Why the pattern would be the reverse in boys and in girls, I really have no idea. And I have no interpretation about the depth of the central sulcus, and even the central sulcus was not our a priori hypothesis. Now I should disclose that the central sulcus was the control sulcus in our study. In fact, the sulci that we were investigating were the Sylvian fissure and the superior temporal sulcus, and there we found nothing. So we're in a kind of conundrum where everything that we hoped to find we did not find, and we find a very strong, what seems to be a strong result in the control sulcus, and we're wondering about how to report that. And of course there's the PhD of a student that depends on this.
Well, in light of the fact that dyslexic males are like females, that's immediately sellable to the press. That's an obvious press release.
Yeah, I don't want to sell that story.
Chomsky would have already predicted this.
Oh, really?
Well, if you say that, then of course.
So anyway, if you have any idea that would facilitate our publication of this, I'm happy to hear about this at dinner.
In what way is the hypothesis that you were going to see effect in one part of the brain a more important hypothesis than you were not going to see it in another part of the brain?
This is dyslexia, and we have good reasons to expect things in the language regions, and so that's why we
But by extension, you had good things to not expect it in some other region.
No,
but disproven that hypothesis.
Yeah, no, I agree. We don't have strong reasons not to expect something in the central sulcus, but not more in the central sulcus than in the para-cingulate sulcus, than in the inferior frontal sulcus. It could be anywhere for that matter. So
those are hypotheses that follow for future studies.
We could always make up a story, but
I'm not saying make up a story. I'm saying you're discounting the result, but you had a strong hypothesis, which was that this is a good control structure, and you have good reason to not doubt it. Why is that? In what sense is that a weaker result than had you found the result that you originally had?
Well, for one thing, if I had to correct for the 3 sulci, for the fact that we tested mean depth, maximum depth, surface of each sulcus, 2 hemispheres. If we corrected by everything, even that result would not stand multiple correction.
So you should correct everything.
Yes. If we report the 3 sulci together, yes, we should. Of course, the temptation is to publish the central sulcus by itself. So we haven't taken any decision so far. I'm happy to hear about your views later. Let's look the other way around. So mapping structure to function is very interesting. Mapping structure to genes is very interesting too. Some people have started doing that, picking on the genes that have been associated with dyslexia, such as these 3, DIX1C1, DCDC2, and KIA0319, and looking whether the SNPs that are associated with dyslexia may also correlate with some brain phenotypes. And so this is an example from the Karolinska Institute, where they have found that some SNPs of these 3 genes correlate with white matter volumes in these posterior, parietal temporal regions in the white matter. with a pretty decent sized group. But of course, yeah, I'm not sure this is the kind of result that can be trusted. I mean, there are so many things that they must have looked at in the brain, and they report only this. But so again, I mean, people are trying to move in that direction, but I think this study illustrates that it's very difficult to produce something convincing, especially when you think of the typical effect size of genetic effects. I mean, genetic effects on cognition are very weak. Genetic effects on brain structure, I expect it to be a bit stronger, but I would be surprised if they could explain more than a few % of the variance, right?
Yeah.
Simon?
Can I say something in relation to that? Because we're part of a consortium that has now about 30,000 different brains, and they're all analysed. I think you're also part of this Enigma consortium. But the idea is that you could actually start to do genome-wide scanning for, say, cortical thickness and volumes of different structures. And what we see when we do that is that the prediction that effects on brain structure should have larger effect sizes than behaviour have turned out to be completely wrong. So we know now.
Really?
We know from GWAS studies that the top hits that you get are still explaining a tiny percentage of the variance, which unfortunately means that when you have even a study sample of 76, you have enough power to test a few SNPs, and then you have to be very lucky to hit on the right SNPs. And interestingly, the Karolinska group have published quite a few papers now on this data set, and they always seem to find something somewhere in different places. So I'd like to see this stuff replicated by different groups.
Yes.
And we failed to replicate some of their results, which we haven't.
Now, okay, it's interesting that you mentioned this study with 30,000 brains where you find smaller effect sizes for associations with brain structure than with cognition. I mean, one possible explanation for that is that the brain measures that you're trying to associate with genes are the wrong ones. And this is one of my next slides, so I'll be coming to that. Here's another example of a genetic imaging study of gene DCDC2, another one associated with dyslexia. And so some people have a deletion, and that's the risk allele for dyslexia. Other people don't have the deletion. And so, of course, the deletion is pretty rare. So here they only have 13 people who are heterozygous for the deletion. And you can see the extent of the differences that they find here. You bet that they don't have a very stringent correction for multiple tests, right? So I think we can very easily disregard this study. Now I'm going to show a study from your group. So this is the study where you have analyzed the plenum temporale in association with a number of genes, in fact in the whole genome association study, right, if I'm correct. And also looking at the sex difference in the asymmetry of the plenum temporale. So here this shows the cluster of gray matter volume that is associated with what gene? With the set of genes of steroid hormone receptor activity in males on the left and in females on the right. And this shows the sex difference in plenum temporale asymmetry, with the females being the solid line and males being the dashed line. Now this study is absolutely great in terms of sample size. Definitely there's nothing to say about this. However, I would be more skeptical about the nature of the brain phenotype that is measured here. And I know that you share exactly because it's stated in the paper that you are perfectly aware of the limitations of this. The issue is that in order to measure the plenum temporale and to compute its asymmetry in such a large sample size, obviously you didn't do like my PhD student and drawing all the plana hemisphere by hemisphere manually. You can't do that on 3,000 subjects, so you use an entirely automated method which is like voxel-based morphometry, where you have to warp all the brains into a common template, voxel by voxel, and then in that common template you use a probabilistic atlas of the plenum temporale, which is shown here, in order to infer the probability that each voxel of each subject might belong to the plenum. And it is on that basis that you compute the size of the plenum and its asymmetry and its sex difference. And so, yeah, I think this is really a very wrong way to measure the plenum. I mean, there are many things that are wrong here. I mean, I object generally speaking to the very notion of mapping all the brains to a common template voxel by voxel, because basically you may be mapping one voxel of one gyrus from one subject to a voxel of a different gyrus from a different subject, so you have absolutely no guarantee that you're talking about the plenum temporale. It's just completely blurry. In fact, we can, yeah, no, I don't have the picture here. And you're basing it, and then in the common template, you're basing it on a probabilistic atlas that has been determined using criteria that are probably not the right ones either. Again, it's a conundrum, because this is the best thing that you can do with such a large sample size, but at the same time we have good reasons to think that it's not a good measure of the relevant brain phenotype. And I think we can even see that if we look at the asymmetry measures here. Like, they are all positive, these asymmetry indices, which is strange because some people have the reversed asymmetry pattern, so we should have people in the negative range here. And I think here, the sex difference that you obtain is probably an underestimate of the real sex difference. It should be stronger than this if the measure was correct, at least according to, well, we can talk about that later. But so, all this to say that ideally, what we would like, I mean, we would like to do some large sample studies just like you have done. But ideally, we would like to image directly the cytoarchitectonic areas. That would be the best possible thing, okay? Or to obtain a reliable estimation of those cytoarchitectonic areas as defined by any relevant criteria, perhaps the receptors. I mean, either the analysis of the cells, or the analysis of the receptors, or the metabolites, or whatever, as we have seen yesterday. Unfortunately, at the moment, this is not available. I mean, you can image some densities of receptors using PET to some extent, but it's very inconvenient and limited. You can do some things using proton spectroscopy in MR, but again, that's very limited to certain metabolites. The resolution is very poor, and so on. If one day we could directly image the receptors using a new imaging technique, that would be the best thing. What we have, either small sample sizes, and in those small sample sizes we can actually measure the right brain phenotypes, or large sample sizes, and in those sample sizes we can only make a very coarse inference of the cytoarchitectonic areas, or the receptors, or their metabolite densities. So this is not a very good situation, I would say. We've seen the pictures from Nicola, which, I mean, this is really wonderful work, and this is the sort of pictures that I would see in the brains. I would like to see in the brains of all my subjects. Unfortunately, it can only be obtained post-mortem. So this TE 2.2. This is the plenum temporale. So this is why I would like to see it. The one thing that I could do would be to try and use their probabilistic atlas to map to the brains of my subjects. The problem is that their probabilistic atlas, I mean, there are several problems. One is that it's based on only 10 subjects. So I would say that the error bars around the boundaries between the regions must be pretty large. These were old subjects. I have children, so we don't know how comparable they are. And then the probabilistic atlas is produced again in a voxel-based manner, using mapping to a common template voxel by voxel, which is again, I think, very, very debatable. So for this reason, I'm not sure that it would be such a good move for me to try and map to this probabilistic atlas. Maybe this will illustrate the issue a little better. This is the pipeline that the images, the T1 images, go through if you want to apply a volumetric probabilistic atlas for brain regions. So you have, this is the original image, you separated it to gray matter, white matter, and CSF, and then you map it to pre-established templates for white and gray matter. And so you can see that the brain has been warped. This brain is slightly like twisted, asymmetrical, and now it has been rendered perfectly symmetrical. So this warping process actually removes some of the neuroanatomical variability that actually may be of interest to us when we study neuroanatomical variability. And it is on this sort of maps that the probabilistic atlases are produced. So the brains of the patients that have been dissected, they are warped to this common template, and if I want to use that atlas, I have to warp my brains to that common template. If you want to do VBM, you further smooth and you work on those images. I mean, you start with such beautiful, high resolution, high contrast images, and you do all your analysis with such crappy things. Okay, when I see those images, I think, yuck. So I think maybe this gives you the feeling why VBM studies do not converge on much. I mean, except like if you study Alzheimer's disease and you have major degeneration of the gray matter, yes, of course this is going to work, but not in a disorder like dyslexia. So what better can be done? I think surface-based mapping is a bit more promising. Remember, this is just what I explained when we did the cortical thickness study. You reconstruct the two surfaces of the brain, and you model them using a very fine mesh. And so the people who produce the algorithms for the analysis of brain surfaces have also produced a number of atlases. And those atlases are mapped onto either onto a sphere or onto an inflated brain like this one. And the good thing about this approach is that when you map, you're still mapping your individual brain onto a common template. But the process of mapping onto the common template respects the anatomy in the sense that it tries to align the main cell site, which cannot be worse than mapping voxel to voxel without knowing where you are. You don't agree?
But I mean, let's say we have seen the result of the cytoarchitectonics. They don't, so the so-called landmarks are not necessarily the boundaries of the cytoarchitectonically relevant areas. So that is a bigger problem.
So
not necessarily, of course.
Mapping is very good and it does produce beautiful results. But, and of course, if you're going to map the central sulcus to the central sulcus, but there are cells that exist in one brain and not in the other brain. And even if you inflate the brain, so what are you going to do with the Jensen sulcus on the hemispheres in which it is present? What are you going to warp it to on the, on the hemispheres in which it's not present?
I agree with you.
Are you going to slice your globe and have a little hole?
Yeah, in fact, the warping, I mean, the surface-based warping cannot take into account all those minor cell sites that exist in some subjects, not in others. It can only take into account the major cell sites, such as the lateral fissure and the central sulcus and the STS and so on. But I think this is already better than the voxel-based approach. At least you know that I mean, like if you study the planum, if you're 1 millimeter above, you might be in parietal cortex, and this is complete nonsense. So the VBM approach is not possible. At least if you align on the lateral fissure, you know where you are. This can only be better.
You can also do landmark constraints, voxel-based
Really?
alignment. And it's not that bad. It's not as good maybe as surface-based, but it's not, you know, a voxel that's in the calculum sulcus in one brain doesn't end up in the central sulcus in another brain, when you warp it to another brain.
Yeah. I mean, there's no magical one, for sure.
No, that's the problem.
Simon?
Is there something special about the plane temporally? Because what we encountered when we started working on it was that different experts who claim to be experts in defining the plane temporally have different opinions about where it should be. So you're talking about Obergurgl has a very particular definition that has to be traced out. And as you said, what we need to have is a system for being able to scale this stuff up. So we go with one expert advice from one set of people, and then of course you face reviewers who say, no, this isn't how you define the brain as a problem. So I don't know, it seems that, say, for the problem areas, there's some clear ways of figuring out really what the most right answer is for from the psychology.
Yeah.
But is this, I mean, does this apply to multiple brain regions of interest?
I think so. I think the work that they have done should allow us to adjudicate between Galaberda and people who have a different opinion about the boundaries of the plenum. And once the data have shown what the best criteria are, then we need to just find the most reliable templates in which to put the probabilistic atlas and then to map the individual brains. That's all I'm saying.
And interestingly, we had a grant to actually try and spend time on figuring out how to define these things and how to bring in data from Carlsbad.
But maybe you're already trying to produce a probabilistic atlas in a surface-based template, right? Because it's
there are collaborations going on with Bruce Fisher, yes. It's not my line of expertise.
But that would make sense. So anyway, I think, I think I'll stop there. I've raised a number of issues, which I think are pretty important if we really want to understand what's brain structure, how it varies, and what those variations explain. And so, I think these are important issues for dyslexia and for disorders, and for also normal variability in cognition in general. And so we can go on with the discussion. Thanks for your attention.